All right, lesson 23.3, all about our stems now. Again, our goal is the same as before, relating the structure and function of each of the major plant organs and tissues to physiological processes. Moving on to our objectives, describe the main functions of stems and also contrast the primary growth and secondary growth in stems too. So let's look at the functions first. While choosing some items on the salad bar, you added some slice. Water chestnuts, some bamboo shoots, asparagus, and a potato to your plate. These things all come from plants, but can you think of something else that they might have all in common? They all come from the same part of the plant. Do you have any idea which one? Yes, we're talking about stems. So then our first objective, describe the main functions of stems. So they all are stems. Stems vary in size, shape, and method of development. Above ground stems have several important functions. Stems produce leaves, branches, and flowers. Stems hold leaves up to the sun, and stems transport substance throughout the plant. Stems make up an essential part of the water and mineral transport system of the plant. The xylem and the phloem form continuous tubes from the roots of the stems to the leaves, and these vascular tissues allow the water, nutrients, and other compounds to be carried throughout the plant. In many plants, stems also function as storage and aid in the process of photosynthesis. For example, desert cacti have thick green stems that carry out photosynthesis and are adapted to store water too. Stems contain the three types of tissues, the dermal, vascular, and ground tissue also, and they are surrounded by a layer of epidermal cells that have thick cell walls and a waxy protective coating. You can see on the outside of this one, epidermals right there. These cross sections through a monocot and a dicot stem show the epidermis, vascular tissue, and the ground tissue. The ground tissue is white, the vascular is a little red, not red, but turquoisey colors there. Same on this side too. Growing stems contain distinct nodes where leaves are attached, all right, as shown in this figure. Small buds are found where leaves are attached to the nodes, little buds right there. And the buds contain the apical meristems that can produce new stems and leaves. In larger plants, stems develop woody tissues that help support leaves and flowers. In the monocots, clusters of xylems and phloems tissues called vascular bundles are scattered throughout the stem, as shown in the cross-section on here. In most dicots, though, and gymnosperms, vascular bundles are arranged in a cylinder or ring, as shown in the cross-section right here. So know the difference between monocot and the dicots, please. In monocots, the clusters of xylem and phloem tissue, called vascular bundles, are scattered about through the stem, and the monocot stem shows the epidermis, which encloses ground tissues in the vascular bundles. The vascular bundles, as we said before, are scattered about throughout the ground tissues. The ground tissue, therefore, is fairly uniform, consisting mainly of the parenchyma cells. The young dicot stems have vascular bundles that are generally arranged in a ring-like pattern, like we said, as shown in this cross-section right here. The parenchyma cells inside the ring of the vascular cells are known as pith, while those outside form the cortex of the stem. These tissue patterns become more complex as the plant grows, and the stem increases in diameter too. So the main objective of the main function of the stems is above ground stems have several important functions. Stems produce leaves, branches, and flowers. Stems hold leaves to the sun. The stems transport substances throughout the plant. But none of those monocot and dicots. Objective 2 is to contrast the process of primary growth and secondary growth. So, unlike animals, the growth of most plants isn't precisely determined, but plant growth is still carefully controlled and regulated. Depending on the species, plant growth follows general patterns that produce the characteristic size and shape of the adult plant. A plant's apical meristems at the roots and shoots produce new cells and increases its length. This growth, occurring at the ends of the plant, is called primary growth, as you can see here. Primary growth, primary growth, primary growth. It takes place in all seed plants. As the plant grows larger, the older parts of the stems have more mass then to support and more fluid to move through the vascular tissues. As a result, stems increase in thickness, which is known as secondary growth, as you can see right here. The figure below illustrates that of a dicot. Secondary growth is very common among dicots and non-flowering plants, such as pines, but is rare in monocots. This limits the girth of most monocots. Unlike monocots, most dicots have meristems within their stems and roots that can produce true secondary growth. This enables them to grow to great heights because the increase in width supports the weight. All right. In conifers, the dicots, secondary growth takes place in the meristems, called the vascular cambium and cork cambium. Vascular cambium, cork cambium. The vascular cambium produces vascular tissues, increases thickness of stems over time. The cork cambium produces the outer covering of the stems. Once secondary growth begins, the vascular cambium appears as a thin, cylindrical layer of cells between the xylem and the phloem of each vascular bundle. Divisions in the vascular cambium give rise to new layers of xylem and phloem. Each year, the cambium continues to produce new layers of vascular tissue, causing the stem to become thicker and thicker. Most of what is called wood is actually layers of secondary xylem produced by the vascular cambium. As woody stems grow thicker, the older xylem near the center of the stem no longer conducts water and becomes what's called heartwood. Heartwood usually darkens with age because it accumulates colored deposits. Heartwood is surrounded by sapwood, which is active in fluid transport and is therefore usually lighter in color. When growth begins in the spring, the vascular cambium begins to grow rapidly, producing, right here, vascular cambium, producing large light-colored xylem cells, resulting in a light-colored layer of early wood. As the growing season continues, the cells grow less and have thicker cell walls, forming a layer of darker late wood. This alternations of dark wood and light wood produces what we commonly call tree rings. Each tree ring has a light wood at one edge and a dark wood at another edge, making a sharp boundary between the rings. Usually a ring corresponds to a year of growth. By counting the rings in the cross-section of your tree, you can estimate its age. The size of the rings may even provide information about weather conditions. Thick rings indicate the weather conditions were favorable for tree growth, whereas thin rings indicate less favorable conditions. In a mature stem, all the tissues found outside the vascular cambium, make up the bark, as shown in this figure. These tissues include phloem, the cork cambium, and the cork. So know that. And as the tree expands in width, the oldest tissues may split and fragment. The cork cambium surrounds the cortex and produces a thick protective layer of waterproof cork that prevents the loss of water from the stem. As the stem increases in size, outer layers of dead bark often crack then and flake off the tree. So our objective 2 was contrast the process of primary growth and secondary growth in stems. Primary growth in stems is the result of elongations of cells produced in the apical meristem. It takes place in all seed plants. In conifers and dicots, secondary growth takes place in the meristems called the vascular cambium and the cork cambium too. So our objectives again are to describe the main functions of stems and contrast the primary growth and secondary growth in stems. Again, both of them definitely help support our goals. And there's a cute picture for you.